All right, let's get started then. Welcome everyone. Thanks for attending this symposium. Yes, I did unmute myself. We entitled this symposium From the Cabinet to the People, where the idea is to share experiences of how the specimens that we curate in collections are used in a museum setting. I'm Jennifer Giron. I'm the acting collections manager of the invertebrate zoology collection at the Museum of Texas Tech University. And the idea of this symposium came to be by chatting with my co-organizer, Alisa DeWell. Hi, Alisa.
Hi. Hi, everyone.
She's a master's student on the Heritage and Museum Science program here at Tech. Alisa started working at the collection earlier this year, digitizing our bees. And very quickly, she became interested in how all this information that she was carefully entering in the database could reach the public. And then as a scientist, I was able to explain how this information can be used for research, but the aspects of outreach, exhibits, and educational components, which are fundamental for museum professionals, are up to her to explore. So we are turning to the ECN community to learn and share ideas about how entomological collections get incorporated in the public-facing side of the museums that house them. I would like to remind everyone that this session will be recorded for later viewing. So please direct all your questions to the speakers using the Q&A feature found at the bottom of your screens. Ideally, do not post your questions to the chat if you can use the Q&A. Also, please remember that the ECN Code of Conduct applies to virtual events, so that inappropriate behavior or misuse of the chat feature will result in people being removed from the meeting or the chat function being disabled. I'll also take this opportunity to end of this session. Hello?
You cut out there for a second, but we can hear you now.
Okay, yeah, my connection at home is not ideal, but in any case, if I drop off the call, Alisa can cover for me. So we will start in 1 minute, and we can welcome Vicky Xuan from the Biodiversity Collections at the University of Texas at El Paso with her talk, Tiny Tunnels, Big Connections, Making Ants Larger Than Life at the Centennial Museum.
Hi, my name is Vicki, and I'm the collections manager at the UTEP Biodiversity Collections. We recently closed our exhibit, Tiny Tunnels, Big Connections, this summer, and I thought I would share some of the strategies that we employ to share our love of insects and get people to understand why studying them was so important. So if you came to my talk last year, you'll know that we had a grant to digitize and organize our ant collections. In total, there's about 230,000 specimens of ants throughout our collections, mostly concentrated in Central and South America. And we got about half of that organized and digitized. There's about 1,600 species, 1,600 taxa throughout that collection. And as a part of this project, we also got these beautiful high-resolution photos thanks to our partners at Northern Arizona University. Now, part of this project was to create an exhibit celebrating the digitization of this project and also the ant collections themselves. And so we started thinking about the exhibit as soon as we could. And at the biodiversity collections, we do a lot of outreach because we don't have any public exhibits. And we use the outreach events to gather some intel on what people thought about the insect collections and the ants specifically. So we noticed that there was a lot of basic knowledge of ants missing, so that was something that we needed to definitely introduce before we got into anything more fun with the ants. And also, people tend to think that ants are only 1 or 2 species, so they might say that there's black ants and red ants, and that's pretty much all they know about them. But the good thing was that patrons were excited every time they saw us bring a box of different kinds of species of ants, even if they thought that they might be pests sometimes. So there was a lot of room for people to want to see this ant exhibit. So we came up with a couple of goals for the exhibit. One, we wanted to challenge some misconceptions about ants and improve that human-ant relationship, demonstrate the incredible biodiversity of ants because there's so many of them that we could choose from, and show off the breadth of our ant collections, especially because we suffer from an extremely low profile. And so this was a really big chance for us to get our name out there and also show how cool and amazing natural history collections are. We thankfully had the assistance of a consultant, Sean T. Dash, who is a former student of Dr. William McKay and has done a lot of outreach and has talked to lots of people about ants. And so he had a lot of experience with science communication and thankfully lent his expertise to this exhibit. And he had this fantastic idea to center the exhibit theme about relationships. Because as he pointed out, ants are ecosystem engineers and they play a role in the network of organisms. So an exhibit about ants itself would be awesome, but showing off how they fit into into ecological communities really will show how important they are. And in addition to that, it would give us a chance to show off all of our collections and show how they can be integrated to produce a singular exhibit. Now, I'm sure all of you are familiar with the challenge of exhibiting insects, namely that they're very small. And to show you the magnitude of the problem, I've measured out this icon, which, if you don't know, human heads are around 22 centimeters, or that's what I read on Wikipedia. And ants, on the other hand, in my experience, are around about 1 centimeter. So I've just learned that human heads are 22 times the size of ants. And so that creates this really big problem because we want to center specimens, but magnifying them might be a bit of a problem. So I wanted to show you how we solved this problem in a couple of different ways and also made ants relatable, since just because they're really small, people don't tend to notice them and it's hard to get to know them otherwise. This is a picture of what the exhibit generally looks like. We try to cover a couple of topics. Some ant basics, symbiotic relationships form the center of our exhibit, but we also featured predator-prey relationships as well as how UTEP collections fit into the rest of the world collections. So I'm just going to show you a couple of things, but our exhibit is currently now online and you can explore a lot of the really cool things that were in the exhibit because I had to leave a lot of things out. So one thing that we did throughout the exhibit was situate specimens in dioramas. And on all of these cases that are clear cases so that little children could look into the exhibits even if they were very short, we used magnifier sheets that were tied to the cases so that people can move around and interact with all of the cases. There's also little magnifying glasses on choice specimens so that people could take a closer look at structures and also examine certain features of insects closer. Throughout the exhibit, we also used digitization products. So this is an example of where we explained what casts were and how to tell the difference between those casts. And so those pictures right there are actually from our paratype photos. And by using multiple species throughout the exhibit, we could explain the same concept and demonstrate variation because, as you know, things don't look the same across species. So this gave us a chance to tie the concepts together. As I said, it was really important that we actually use real specimens and not just images and prints because, as you guys know, scientists learn about species from specimens themselves. And so by using real specimens, we can give patrons a chance to learn in the same way that we do and bring that scientific process closer to them. And so really we wanted to situate specimens front and center to learning. So this is one of the ways that we did so. This display also was put together by one of my students, Alonzo Corral. He's an undergrad, and he picked out all these specimens after learning how to sex them and tell the difference between those casts. This is another example of how we situated specimens in dioramas. Sean gave us this great idea of a little restaurant kind of idea where we situated the prey in the center of this plate and had predators on the outside of the plate to express predator-prey relationships. And so we chose animals that specialized on ants, for example, like horned lizards, and this gave us a chance to bring in multiple species that have similar relationships to show how these organisms provide these services for very, very large organisms. And then, of course, we also used interactives through this exhibit. So this is one example. We had a poll where we asked patrons to think of a relationship that fit under one of these categories and stick a sticker appropriately. And so even though we weren't able to get through this entire exhibit because of COVID, this particular interactive was very popular, and it was the one interactive that we could collect some data. So it's really great to know that El Pasoans really love to have positive relationships, but it's always fun to see some of the commensalistic and parasitic ones. We magnified labels in order to allow patrons to kind of get a sense of what the labels on all of these specimens were. And so that way the specimens were just there because they exhibited the species, but also gave a sense of the span and time and place of where these ants came from. So very curious patrons could take a look at that. We also tried to put the cases in giant diagrams themselves, and so we featured some big vinyl pieces like this Cecropia tree, in which patrons were transported to Central or South America, where they could take a peek inside of this tree and look at how ants are have this mutualistic relationship with Cecropia trees. And so it kind of felt like patrons could look inside the tree as well as pair it right next to their barium specimen so you could see how large these ants were in comparison to the tree itself. The Centennial Museum also has a relationship with a local company, Augment El Paso, and they augmented my students' diagrams of a particular commensalistic relationship where birds pick up ants and provoke the ants into stinging them for a reason that we're not quite clear yet. Her diagram shows what's going on with the birds, but this augment reality did bring this entire diagram to life. So it really pairs the specimens with the behavior. Now, that bird right there is not the only example of this relationship happening. We tried to get multiple examples of anting behaviors, and that way we could show these extremely large animals depending on these tiny little ants. And so instead of shying away from the fact that ants were so tiny, we tried to even contrast that size, contrast the size issue. Of course, like other museums before us, we used high-resolution images from local stories and also from collaborators. My student Veronica Gomez had cataloged this new spider that had been discovered at the local research station, and so we could show some of the really neat relationships that happen even within the Chihuahuan Desert. One feature that we had been thinking about from the beginning was 3D prints. And unfortunately, our 3D printers were broken locally, and so we had to, our 3D scanners were broken locally, so we got donations from the Kanamo Lab in Japan and also the Lucky Lab from the University of Florida, and we used 3D prints in a couple of different ways. So one, we had them paired with the specimens themselves, these full body prints, so that you could get some real cool detail on the morphology of these ants. And then we had these head trophies that we could use to show off the really cool biodiversity of ants that we may not have, but that the University of Florida had. And finally, we use these prints to show off head morphologies and do a little bit of functional anatomy. So in the case where we talked about predator-prey relationships, we could focus on these really large prints of head since we didn't need to print out the whole entire body. And finally, here's a great example of how we used our digitization projects to even design display cases. So again, Sean had suggested that we have a case of a bunch of ants through the Chihuahuan Desert, but because we didn't exactly know inherently what ants were in the Chihuahuan Desert, my student Muriel Norman created a map of the Chihuahuan Desert, found the records that appeared within those boundaries, and then created a visual and physical checklist of the Chihuahuan Desert ants with this display. And having everything organized now, we could quickly go through the collections and pick out what we needed. And so that just shows some of the neat examples of how we displayed specimens throughout the exhibit. What I do know is we had about 1,000 visitors in the first month of the exhibit opening before the shutdown of COVID, and we ended up displaying about 150 specimens in total. That's including the relationship partners, and featured nearly 100 species of ants, which is really great. This is just a smattering of where the ants in the exhibit came from. Because we have everything now in our collections management system, we can really pull this data together. And as you might have remembered from my first slide, this does cover the breadth of our collection. So I think we did a good job of showing off a good sampling of what we have. Gotta say that having an organized collection of ants really makes putting exhibits together a lot easier. We can quickly search and see what we have that might fit particular stories. And doing so and being a part of this exhibit, we now can take these exhibit pieces piecemeal to our outreach events, even though the exhibit is over. The exhibit is now online at the Centennial Museum website, so please feel free to explore, and because I didn't get to talk a lot about other products that were in the exhibit, and I'm happy to answer any questions about the process. Thanks.
All right. Thank you, Vicky. That presentation was amazing. It blows my mind, many things that you were able to do with your specimens. And if people have questions, please direct them to the Q&A. For now, Karen has a question. So Karen, go ahead.
Hi, Vicky. I was just curious about those tiny little magnifying glasses that you had on the display. Did you buy those pre-made or did you make them?
Yeah, they were pre-made. They're actually on Amazon and they worked really great. So I think they magnified them by about 1.5 times. So of course not perfect, but at least it gets people leaning in and interacting with the specimens themselves.
Thank you. I have a question. You use several different types of display methods. Did it take too much customization of the furniture of the exhibits to be able to achieve the different things, exhibitions that you did?
Yeah, so really the only thing that we built were some framework to have the specimens jut out at an angle, so those particular cases, but it's really just one type of case. So our preparator made those frames that fit those cases. So that wasn't really difficult at all. And then we found that the foam, we just had foam board and paper that was mounted on the foam board to create all those different dioramas. And one thing I did find was that the paper that we were using is pretty hard. So we had to be very careful when we were pinning the specimens to the foam boards and just kind of very slowly wiggle the specimen in. So it's not as easy as when we're using regular foam for collection management. We can just stick it right in. You need to be careful, otherwise you'll start flinging specimens all over the place.
Yeah, one more question I have is of the approaches that you took displaying the specimens and the information associated with the specimens, which one do you think was most effective to get people interested into the life of it?
Gosh, it's kind of hard. I really felt like, I mean, we only had really 1 month with everything. I feel like people really enjoyed the predator-prey relationship display, especially because people usually think of ants as only eating sugar and that they're in their kitchens. And we got to show off all the things that ants eat, and they specialize in different things. And plus there was so much diversity with that, with interactions, that I think that display was probably a lot of people's favorite.
Good, good. Do we have any questions from the audience? Ashley comments that she loved the idea of displaying specimen labels.
Yeah, I wish we could even do more of that. Of course, we have limited space. And yes, I think it would be also really great at some point to have like a penning demonstration with giant labels.
Yeah, and I guess it also kind of makes people pay attention to the little specs that they are looking at and how they come from different places or have different information. So I really liked how you explored the different ways on how people can get in touch with what the ants do in the ecosystem. Thank you so much, Vicky.
Thanks. I noticed that Greg mentioned that he used the next larger pen. I think that's also a really great idea. I don't think we thought about that beforehand, so we were just really careful.
All right, I think we need to get going for the next presentation now. Thank you so much, Vicky.
Thanks, guys.
Okay, so next up we have Alisa DeWell from the Museum of Texas Tech University, and her talk is entitled Expanding Access, Creating Outreach Tools for Entomological Collections.
Hi, everyone. Welcome to my presentation, Expanding Access, Creating Outreach Tools for Entomological Collections. My name is Alyssa DeWale. I'm a Heritage and Museum Sciences graduate student at the Museum of Texas Tech University, and I specifically work in the Natural Science Research Laboratory. So a brief outline of the presentation. I'll go through a brief introduction, followed by some background on the Museum of Texas Tech University, background on the invertebrate zoology collection, followed by some digitization to improve accessibility, and then digital content for outreach and education, followed by the conclusion, and then some acknowledgements as well. So here's a photo of me so you all can see what I look like. Before arriving at Texas Tech University, I received my Bachelor's of Science in Anthropology from the University of Tennessee in Chattanooga. This is where I initially explored my love and passion for museums, which ultimately led me to the Heritage Museum Sciences grad program at TTU. At the beginning of this year, I began working with Dr. Jennifer Gueron in the invertebrate zoology collection as a student assistant. And ultimately through this presentation, I'm going to explore some of the ways in which outreach tools can further expand access to entomological collections in a museum setting. So the Museum of Texas Tech University, it was founded in 1929. Exhibits throughout the museum, there are 9 permanent galleries with 7 for traveling exhibits. Some of the current exhibits on display include art, quilts, paleontology, Southwestern Native American art. There are currently 6 collecting divisions as well. These include art, clothing and textiles, anthropology, history, paleontology, and natural history. And collectively, the museum has roughly about 8.8 million objects in its holdings. There is also the Lubbock Lake Landmark, another division of the museum. It's an archaeological and natural history preserve, as well as a dig site at the northern edge of Lubbock, Texas. And finally, the Natural Science Research Laboratory, or the NSRL, has about 5.5 million objects, which actually makes it the largest collection or largest natural history collection in the state of Texas. Collections include mammals, fluid preserved, birds, radioactive, invertebrates, and genetic resources collections. the invertebrate zoology collection. It houses about 4.6 million specimens from around the globe. The collection primarily consists of insects and crustacea, medical veterinary specimens, and arachnids. We currently maintain our collections data online. So through the Symbiotic Collections of Arthropods Network, or SCAN, and the Global Biodiversity Information Facility, known as GBIF. But primarily, most of my work and what I've been doing has been going right into SCAN, so that's what I'm personally familiar with. And next, some information on digitization to improve accessibility. So I initially got started in the invertebrate collection through a bee digitization project, which I'm still working on. This initially began with entering each specimen's label data individually into SCAN. We then implemented an optimized workflow by utilizing a website called Digit Specimen, and in which we would take dorsal view images with labels, like you can see here to the right, and Digit Specimen uses AI technology to read these text labels. The AI can then convert this information that's extracted into Darwin Core Standards, and after that information is reviewed, it can then be extracted into an Excel sheet. Once this Excel sheet is extracted and downloaded, you can then manipulate that, upload it into SCAN or any other database. And I know that this is way too small probably to read, but here's a poster that Jennifer and I actually presented at the Mountain Plains Museums Association conference a few weeks ago on this new workflow. And if anyone would like to receive this, just let myself or Jennifer know, and we'll be happy to share it with you. And through this project, I've been gaining additional skills like learning Darwin Core, georeferencing, paying really close attention to detail, photographing bees and specimens and such as well. And I've even learned a little bit of some bee morphology. But so through this project, I've really questioned how more accessible this information is going to be to the public. So this is amazing for researchers and will be useful, across the globe. But what about everybody else? So I think as museum professionals, we need to really consider this when working through these types of projects. And now to get to the nitty gritty, to really get into the digital content for outreach and education. So because of my interest in the accessibility of collections, I've sought out a project and some experience with the education department at the MOTTU so that we can make some of this data on the bees and other pollinators more available to our public. So something that I feel like might be kind of basic for museums and education programs is just making this educational content available online. So that would be, creating lesson plan activities for different age groups, providing short readings, activities, outdoor activities for school groups, families, but also homeschool children and even visitors, not just schools and teachers. It seems that through my experience at the MOTTU that a lot of the public doesn't really seem to realize to what extent of the natural history collections that we have here. And so I think that by collaborating with the education division to create some new content, this can really help spread the word and this new knowledge with the public. So we should always really be considering how and what we're doing is going to turn into educational content or general programming. And so here's just an example above of our website's educators tab of available materials. Some of the tabs that I've looked at don't have too much information, but that's just due to, I think, our education division, working on some new things as well. Next, another great outreach tool that can be used and really beneficial to these types of collections would be 3D modeling, and how also this can really improve the visitor experience all around throughout the museum, but even for those visitors that might be tuning in from their homes. This fall, I've been taking a digital heritage class, which has really brought to light some of these topics on 3D scanning, 3D modeling, 3D printing, photogrammetry, and much more. So I think it's going to be really fun to see, and as we're even seeing now, how these 3D models of insects are going to be created. And even though as we're doing this, still to be really thinking about, how are we going to get these to the public? How are we going to educate the public about where to access these, how to utilize them? And so it can really be a tricky process. So through my class, I've been trying to work on making a 3D model of a jewel beetle. And so we've been using Metashape software to do this. However, these specimens are significantly smaller than other objects that are typically 3D modeled, or so as I've seen throughout my program in our museum. A lot of ethnology type objects are used, a lot of lithics are used to do 3D models. So I've personally been having some issues with that, and I'm currently working through that. And as you can see in that bottom left or bottom right corner, that's not really a full model yet. It's kind of choppy and chunky. So, but yeah, so I think that these 3D models can really create a whole new interactive that can engage also even the younger audience in such an increasingly digital world. And lastly, regarding digital content, another idea that I think that museums and institutions should really take advantage of is utilizing your outdoor spaces. Those spaces that are just outside of your museum, in front, on the side, even parks nearby. It doesn't even have to be a park in walking distance, something in your town or city. This bottom photo on the left is the Arroyo space in front of the museum. And so this can be, if you utilize these outdoor spaces, you could create handouts, educational activities that people can take even on their way out, like walking out of the museum, or it can be content that you provide online that anyone can access at any time. And then in addition to this, and what I'll be doing with Education Division in the spring semester is using the type of beacon technology, specifically with the company Think Proxy. It uses beacons in combination with geographical landscape, or you can even place them just throughout exhibit spaces. So there's a small box, like you can see in this photo to the bottom right. It's weatherproof, so you can place it throughout kind of any areas and it shouldn't get damaged. And then ultimately, when you walk by this beacon, a page or a ding will pull up on your phone, which will provide you with whichever information that you as the museum has associated with this specific beacon. So for instance, my goal with the project is to place about possibly 4 to 6 of these beacons outside of the Arroyo space. And that will include some information on specific pollinators, different types of plants, maybe even information about this ecosystem. And so it's really presenting all of this information to visitors right from their phones. So you don't even really have to go into the museum to still experience almost like a museum type of setting. It's a natural exhibit, you could argue. And additionally, for other free tools could be used like iNaturalist. So these could be used and promoted by the museum. This could allow museum visitors to interact and be involved, even if the museum is closed. They don't even have to really stop by the museum or come in if they choose not to. And it could be a really good way for families and visitors at home to get outside of their homes as well and get outside, especially since we've all been inside for so long, it feels like. And so ultimately, to conclude all of this and close this out, I think that as we continue to digitize and curate these collections, we need to always consider how we'll get this information to the general public beyond researchers. So it doesn't stop there. We've got to just keep going. What else do we need to do to make this understandable and digestible by others? There's also a great opportunity now with so much digitization to take exhibits out of the museum and into the world by utilizing our outdoor spaces and creating engaging activities for our visitors. And that basically results in my presentation, but I would like to go through some acknowledgements. I'd like to thank the museum's executive director, Dr. Pan, Heath Garner, who's the collections manager at the NSRL. I'd also like to thank Dr. Jennifer Giron, the invertebrate zoology collections manager, for allowing me to start this project in January. And she has just become such a great mentor of mine through this year and through all my crazy ideas. Also like to thank Dr. Jill Hoffman, the curator of education, for allowing me to work on this project well into the spring. Also Dr. Stance Hurst, who is my digital heritage professor. And I'd also like to thank the Heritage and Museum Sciences graduate program for giving me the opportunity to study here and work at this institution. Without this program, I really wouldn't be here today and even giving this presentation to you all. And lastly, before I officially end, I'm almost finished with my coursework, and I'm actually open to 6-month internship opportunities that might be starting in May or June of next year. Although my background is in anthropology, I've really enjoyed working in the entomology collection, and I would love to stay in entomology if possible and continue like my passion through this. If anyone is interested in speaking with me, please let me know and reach out to me through email, and I would be more than happy to send my CV, resume with you, answer any questions as well. So thank you guys so much for watching my presentation.
Right, thank you, Elisa. We have a comment in the chat about how intriguing is this beacon technology. Can you expand a little bit about it?
Yes, yeah, just a little bit. So I know the museum got this technology a couple of years ago. And so, yeah, so it's just that beacon and that image. And I understand that you work with, I don't know if it's like a content person on their team, but basically we put together the content, whether that's images, you know, a 3D model to interact with, any type of readable content. And then it's just associated with that beacon. And then when you walk by, it'll pop up. And then I understand too, I think you should be able to then access that when you leave, you know, when you're away from the beacons as well too. So that's a brief.
Thank you. All right, we have also a question by Francisco Sola. It says, could you share the poster on free digitization? And maybe you can also explain a little bit of the process.
Yes, yeah. Yeah, I was trying to find the link. We have an online link, so we can share that in the chat as well. But yes, yeah, through this free digitization, it's called Digit Specimen, and you don't have to make, you know, you don't have to pay anything. It's completely free. You take dorsal images with the specimen along with the labels, and then this AI technology will read the labels, and that's including handwriting. It's not necessarily, it won't pick up the full word, especially with script, but it also has a training model, so you can have it train as you work through reviewing after this AI kind of takes that from the labels. And then after you're done reviewing, it'll download it into an Excel sheet, and then you can kind of manipulate that however you'd like, add things. This is when we do like the georeferencing aspect of the project. And then you can upload this Excel into Scan or another database as well. So yeah.
Yeah, I guess one of the other steps that we've been doing is also that we rename the images, and each of the images gets linked to every single specimen. So it also, like after Elisa made the exercise, it reduced a little bit of time. Like it has several advantages. Do you want to tell them?
Yeah, so it'll, with the renaming tool, it'll rename them all like in a row, but they have to be, I think, in order from the way you took the camera, or else it'll mess up how you number them. But yeah, then that way you don't have to label each image individually. So that also takes out a large step.
And I guess the other advantage is that you have different activities, and it's not just entering data by hand.
Definitely,
but
yes,
you have
breaks it up.
Some time to take pictures, and then some time to upload the pictures into the Digit Specimen website, then download your data, double check that everything is in the right place and with the right spelling, I guess. And then you get to link all your images into with your specimen. So it also allows for less monotony in the processing of this information.
And well, before that, I'll find that link to our presentation, and then I'll put that in the chat as well for anyone who's interested too. So,
all right. Thank you so much, Elisa.
Thank you.
All right. Then it's time to move on to our next presentation. We have Laura Porturas and Andy Deans from the Frost Museum, Frost Entomological Museum, with their presentation, Communicating Through Specimens at the Frost Entomological Museum.
Hi, and thanks for coming to our talk, Communicating Through Specimens at the Frost Entomological Museum. The Frost, just in case you're not familiar, is Penn State University's entomology museum. We are part of the university's College of Agricultural Sciences and care for approximately 1.5 million arthropod specimens. And right next door to our collections is an exhibit space. Here we have approximately 500 square feet, of which we have the freedom to develop material for the public regarding insect biology and ongoing research in our college. In this space, we showcase thousands of objects, including arthropod specimens, entomological gear, historical artifacts, and a photo gallery. One aspect of the space that has really driven the overarching theme of the museum is its proximity to the research collections. They're only separated by a door. Because of this, plus the fact that we're a university collection, we wanted to emphasize not just how awesome and super cool arthropods are, but also how and why entomologists collect and preserve arthropods, and how specimens can be used to better understand the world around us. So, because the layout of our space is circular, we were able to plan and have some control over the order in which visitors are ingesting our material. Assuming visitors walk through the door, move left, and keep going in that direction. So exhibits were laid out in this order: the history of the museum, how and why we collect arthropods, how and why we prepare arthropods for preservation, and what specimens can teach us. The first exhibit visitors see is called The History of the Frost, which marks important time points in our museum's history and notes who was involved in the growth and development of our collection. In the glass case, we display specimens to illustrate aspects of the museum's history, but also to highlight their scientific value. For example, we share through exhibit text that although our museum was founded about 50 years ago, some of the specimens housed here are much older. We explain that these specimens have high scientific value because if preserved well, historical specimens allow us to compare across decades and can tell us about how the world is changing. Alongside this, we've staged a specimen from our museum that was collected way back in the 1800s and is still in excellent condition. The specimen provides proof to visitors that with proper care, specimens can last for centuries. Another example of specimen use in this display is through featuring the work of one of our previous curators, Casey Kim, who was heavily involved in forensic entomology. Blowfly specimens of different life stages are laid out next to each other for visual comparison, so visitors can build the connection in their head of how having specimens like these for reference and comparison can be scientifically useful. In this case, larval specimens of known ages can be used for comparison against blowflies collected from a corpse to estimate the time of death. After visitors learn a little bit about the history of our museum, the next exhibit that they'll come across is Collecting Arthropods. Here they learn not only about the different tools that entomologists use to find and collect insects in various habitats, but also why we collect. The text reads, "To understand how arthropods impact the world around us, entomologists must collect specimens. Specimens allow for close examination of important adaptations and anatomical traits that are unique to each species. Specimens also contain artifacts, pollen, gut contents, DNA, parasites, for example, that inform us about the habitat at the time of collection." Again, this is something that we try to emphasize to strengthen the public's understanding of what exactly collections are used for. The next exhibit, just to the right, is called Preparing Specimens. Here, we inform visitors about how and why specimens are prepared differently for preservation and what information is important to keep with the specimens on the labels. Below our poster guide are physical examples of specimens that have been slide mounted, pinned, preserved in ethanol, and preserved in envelopes. These specimens give context for why some of the specimens that they saw in the History of the Frost cabinet were presented in different formats, on pins and tubes and envelopes, while also reinforcing that our goal is to preserve these specimens for centuries for scientific use. So, in the first room, we try to lay out the baseline for what an entomology collection is for, how insects are collected, and how they're treated for long-term preservation. We try to emphasize that specimens are necessary for learning about insects and that, in part, they're the reason that we know as much as we do about arthropods. The rest of the museum highlights diversity and natural history of insects and also some aspects of the research going on in our department. So after this first room, visitors walk through a hallway where each of the two walls exhibits an awesome array of insect diversity and natural history. The first wall is called Insects of Pennsylvania and highlights the diversity of insects people can see in our state. This is my personal favorite in the museum for a couple of reasons. First is that this display was composed largely from unlabeled trap residues from a colleague's project that didn't have labels or appropriate data and so just didn't really belong in the research collection. We were obviously super happy that we were able to repurpose these specimens and put them to good use. Second, people are often shocked when they see that there are so many different kinds of insects in Pennsylvania, which was exactly our goal. Specimens are clustered based on the order that they belong to, so visitors can appreciate the diversity of all the insects, but also compare and contrast within the flies, within the beetles, bugs, etc., because they're grouped together. In addition to just the wow factor of the Pennsylvania diversity, there are numbers associated with selected individual specimens, so that when visitors look for these numbers on the signs to the left or the right of the display, they can find small natural history notes about the different species. So for example, if someone is looking at this dragonfly specimen, they could check the number, then check the sign, learn what it's called, and a quick fact about the species. Or if someone was looking at this moth, they could check the number, then check the sign, and learn a quick fact about that species. There are 100 numbered specimens total, and this is completely anecdotal, but I've heard visitors say things like, "Oh, I didn't know that. I see this in my backyard all the time," which means to us that these previously junk specimens are being recognized by people in the public and teaching them about animals that they might not have ever taken the time to learn about otherwise. Directly across from our Insects of Pennsylvania wall is a display called The Incredible Diversity of Insects. Here we have some of the highly popular WOW specimens and exhibit insects of all sorts of shapes, forms, and colors. We aim to teach visitors that there is often some sort of biological explanation for the strange appearances that you see in arthropods. And similar to our Insects of Pennsylvania exhibit, selected specimens have numbers that can be referenced on side panels. Through the exhibit text, one of the things that we talk about is how color is formed and what it can be used for. And we display specimens as evidence for the information that we're trying to convey to visitors. So, for example, our text explains Müllerian and Batesian mimicry, and then directly below, we use specimens to illustrate what we've said in text. So we place different species of bumblebees on a map of the US and locations where they can be found, so people can see real bumblebees that inhabit the same regions, have evolved the same warning coloration patterns. Similarly, we flag specimens that are actual stinging insects and place them beside non-stinging mimics so people can compare and contrast their color patterns. We also have a section on this wall that highlights habitat adaptations. The numbered specimens here have information referencing some sort of visible adaptation, whether it be a flattened body, digging legs, pollen gathering hairs, whatever, and we cluster other specimens with similar adaptations around the numbered ones with the goal that people will see the trait of the numbered specimen, learn what that trait is an adaptation for, and then recognize the same trait in the surrounding specimens to infer something about their biology. The rest of the museum focuses on areas of research at Penn State, like honeybee health, pollination by native pollinators, and invasive insects. And so the last example of specimen use in our exhibits that I want to share with you is in our spotted lanternfly exhibit. This invasive insect is particularly important in our region given the spotted lanternfly's feeding preferences and our large fruit industry. The exhibit itself explains what is a biological invasion, what the spotted lanternfly is and how it was introduced, why it's a threat, and what's being done to control the threat. The role of specimens here is by no means groundbreaking, but I think it's important. We've arranged the spotted lanternfly in a nice pattern that draws the eye. But what this does is allow people to become familiar with what the insect actually looks like. Pictures can do a lot, but seeing its real life size, shape, colors is also important. So hopefully visitors in our county, where the spotted lanternfly hasn't quite spread into yet, can better recognize, kill, and report it if seen. And similarly, we've arranged other invasives in a pattern that will hopefully draw their eye so that they might learn to recognize other invasive insects that occur in the area as well. So here at the Frost Museum, we have tried to incorporate specimens into each of our exhibits to help convey to visitors what collections are, how specimens are preserved and used, to share natural history, and to bring clarification to biological concepts. We are constantly tinkering with these exhibits and will periodically be rotating new ones in to replace the old. So we're happy to answer questions and would love any suggestions that you think might help us improve the space or the use of specimens within our exhibits. Thank you very much for your time.
All right, thank you so much, Laura. That was a really nice presentation, and I think we have a bunch of comments in the chat saying how amazing this exhibit look. Like a lot of us are envious and want to go there just to see all of the things that you have available. One of my, well, we don't have
a lot of really nice comments that you guys are saying in the chat section, so thanks.
Yeah. One of my questions is, how interactive is this little station that you have for showing how to mount specimens? Can people actually play with the tools or something, or with the specimens themselves?
Yeah, so we don't have any sort of like interactive piece where they can mount stuff themselves, but we have a microscope there with little signs on how to use the microscope, so they can take the different types of preparations like out of their, like the slide mounted things are out of their container and you can see them underneath the microscope, or the pinned ones are in this little case so the specimens can't get damaged, but everything can be picked up, looked at in their hands or underneath the microscope. It's one of our more interactive displays. So that one's a good one.
All right, thank you. Do you have any way to measure how much people learn from all the things that they get to see throughout the exhibitions?
That was something that I wasn't sure if I could ask as a panelist. So there's the first talk, I'm sorry, I forget her name, the person presenting it, suggested that she was able to collect data from her exhibits. I wanted to ask like what kind of data she's collecting, but no, we don't have any sort of like standardized way to measure that, but we just get feedback from people occasionally. We have a visitor log where occasionally people will sign their name and then write comments, and we'll get some feedback that way, but nothing that we could necessarily analyze.
Okay. And do you have an idea of how many visitors you get well, I guess there's a pandemic going on, but do you have any estimates of how many people walk through during a month?
Yes. So we were closed for a really long time, then opened for a short period, then closed again because of COVID, and just reopened this fall. I think the museum isn't on everybody's radar on campus at this point, but we've been seeing a slow and steady increase in the amount of visitors that we're getting. And it's super variable. So we might get like 5 or so people walking through a day, but then classrooms will come through and there'll be like 50 people in a day. And it's a relatively small space. Our max capacity is 25 people. So if we get 50 people in a day, we're pretty stinking pleased. But yeah.
All right, thank you. We have a question from Floyd. He says, UV protection for the displays to keep the specimens from fading or bleaching from light exposure. Do you account for that?
Yeah, so in the windows that are facing the incredible diversity of insects display and the other ones, those have like clear UV stickers on the windows. We recognize that it's not ideal to have windows right next to these specimens, but we've taken that precaution to try to minimize the amount of UV radiation they're getting.
All right, thank you so much. Okay, so it looks like we don't have more questions. Is there anything else to highlight about your exhibit?
We'd be pleased as punch if you guys came to see it next time you're at Penn State.
All right, thank you so much, Laura. All right. Our next speaker is Nicole Gunter from the Cleveland Museum of Natural History, and she will be talking about dung beetles in the dome, new outreach opportunities for digitized data.
From the cabinet to the people, or from the cabinet to the dome. Today I'll be talking about a milestone for my NSF career grant, the release of new software that brings digitized data to a new audience in the planetarium dome. But first I want to highlight some of the entomology outreach that happens at the Cleveland Museum of Natural History. We are world famous for our viral beetle display, but insects also feature in other parts of our galleries, including the recent dung beetles exhibit and regular public programming in the current science studio. We're currently undergoing a major renovation, and I promise you that there'll be new and exciting insect displays in the new galleries as well. However, beyond the galleries, I envisioned more. I wanted to bring dung beetles to the dome. When most people think about planetariums, they think about the ability to explore the universe, as traditionally they've been used to teach astronomy. The powerful computers behind the scenes project spatial data to the dome, which creates the immersive experience that the audience knows and loves. Beyond exploring space, advancements in planetarium technology now promote the exploration of Earth in high definition. The Cleveland Museum of Natural History, along with thousands of other institutions around the world, are part of a global initiative to digitize their collections. Their data are served to national and global aggregators, such as iDigBio and GBIF, which combined have amassed over 1.9 billion records. My vision was to leverage this publicly available digitized data to transform how we teach biodiversity sciences, not only at our museum, but potentially at other planetariums around the world. There is endless potential for the amassed digitized data, not just for research, but also outreach. Planetariums are powerful teaching tools and underutilized in fields outside astronomy. Bringing biodiversity data to the dome would create new opportunities to explore the importance of natural history collections, research, and also share digitized data with the public. The initial proof of concept that we could view digitized data in the dome formed the educational component of my career grant. I highlighted that while it was certainly possible to display distributional data, it held so much more potential in the temporal stories that it could tell. However, to leverage the full potential of Darwin Core data, we needed new tools within the planetarium software that could handle the temporal stories. Of course, planetarium software design is out of my wheelhouse, so I reached out to Evans and Sutherland's, the designers of the world's leading planetarium software, Digistar. This software is what we use in our dome, and after a handful of conversations, a new collaboration was formed. If my dream of digitized data and dung beetles in the dome was ever to become a reality, then we would need a user-friendly tool for our target audience, the planetarium educators. While Darwin Core is an excellent standard for biodiversity data, it can be overwhelming for those who are not familiar with it. One of the first discussions that I had with the team at Evans and Sutherland's was about Darwin Core data and its importance for the project. Our design discussion focused on 3 goals: leveraging event data to animate change through time, filtering any Darwin Core terms, and answering audience questions in the absence of downloaded data. The solution to the first 2 goals was solved through the creation of an executable that could take a Darwin Core CSV file as downloaded from any data aggregator and transform it into a format that could be viewed in the dome. While the third goal was to answer audience questions, was addressed by a plugin that reads directly from the iDigBio portal, renders it, and projects it. Let's walk through these tools that make up BioViz. The CSV to KML tool generates a filtered file in a matter of seconds. First, the user runs an executable, loads the CSV file, and names the output. The user is then prompted by a series of simple questions that would ask you if you'd like to filter by year, month, or day, and then if you'd like text labels added. Next, there are options on how to color code, with all Darwin Core terms for which there are multiple options available there to choose from. The number of unique values are also displayed. The user can choose color and shape of the icon and can also exclude data groups as needed. Note that if you have a large data set with complex color coding, for many unique values, it may take minutes to go through all the coding options. Once everything has been color-coded, a KML file is saved that can be viewed in the dome. In this example, I color-coded preserved specimens in yellow, human observations in blue, and excluded any unknown records. This executable was also made into a GUI that runs in Digistar 6 and 7. Dan Rees, pictured here, is one of the 2 developers that I worked with at Evans and Sutherland's. The other, John Paul Ownby, were both incredible, to work with. So I just want to give them a shout out here, and full credit goes to them for all of this development. You're probably wondering what it looks like in the dome, and I've recorded some use cases to share. These were captured in Digistar's dome view, which produces a suitable output for a 2D screen. So let's take it away. Engage your imagination and picture yourself in the dome. When seated, the outside of this projected circle would look like the horizon of the screen. Let's start with a tour of the data that first inspired this idea, and an example of how scientists could use this tool to communicate their research within the planetarium. This is a projection of some of the Australian native dung beetles that I study. The data is color-coded and filtered by species. Keep in mind that as we zoom in close to the edges of that circle that represented the peripheral of the dome, it may look a little bit distorted, but within the planetarium, this appears quite natural. So the colored dots that you're seeing here represents 3 species of flightless dung beetles that are found on the east coast of Australia. As we zoom in close to the Earth, it becomes evident that they're restricted to these denser dark green patches of closed forests, and their habitat is separated by large patches of open eucalypt forests in the lighter green. The cities of Brisbane, Ipswich, and Gold Coast are visible on the grey in the east coast, and we can see that there's a yellow species found in the north, one in the south in blue, and a third to the eastern coast in red there. These incredible satellite images in Digistar allow us to explore the topology of the habitat. While exploring stationary dots on maps is compelling for some, the innovation in BioVis lies in our ability to quickly filter time series records, which will allow us to animate biological events such as the well-known migration of monarchs. Here we have filtered over 20,000 records of monarch butterflies by month. Each file is generated in just a matter of seconds and compiled into a loop in Digistar. We can see that the distribution of records changed month by month, with monarchs overwintering in the south before heading on their adventure north in the spring and arriving in the far northern extent of their range in summer before returning on their southward migration in the fall. Next up, we have another magnificent migration, this time of the ruby-throated hummingbird. There are almost 2 million records of this common species. This data is really impressive as it highlights the ease at which BioVis can handle the large data sets, not only when filtering and compiling the records, but also when viewing them within the dome. Here we have sorted the data into blocks of 2 weeks to visualize the spring migration. It's not just migrating species that can tell compelling stories. This data is filtered by year and shows the spread of an invasive species, the brown marmorated stink bug, as it establishes across the US. It's incredible to see that just in a matter of 20 years how widespread this agricultural pest is. Similarly, data could be filtered by year or decade to show trends in biodiversity loss, expansion of ranges of vectors that cause diseases, and many more. Maps like these can be integrated with documentary-style footage and images to create new programs on biodiversity. This next map may look very familiar. The second new feature in BioVis is a plugin that reads directly from iDigBio's portal, projecting it to the dome with a click of the button. Here we've searched for the popular recreational fish walleye. The distribution of the species is pretty compelling and aligns with the ecology of the species. We can clearly see its presence in watersheds and even the outline of the Mississippi, Illinois, and Fox rivers, as well as the high density in the Great Lakes. A keen angler is probably able to spot their next fishing spot. The legend from iDigBio can be juggled on and off, and this tool was created mostly to answer audience questions on the fly by harnessing the power of the iDigBio portal within the planetarium. Finally, the thing that I'm most excited about is our ability to search the records of our own digitized specimens at our institution. When I host tours of our collections, I mentioned that most of our samples are collected regionally, but we have some representation from all around the world. This is hard to imagine as our collection holds millions of specimens. However, the extent of our regional collections becomes much more clear when we search for our museum acronym within the iDigBio portal. This data is from the invertebrate zoology collection and represents the insects that have been digitized so far. The new Digistar plugin, BioVis, was released earlier this year and is available as part of Digistar 6 and Digistar 7 software. 165 planetariums around the world can now easily display digitized data in a compelling way. BioViz brings collections and research to a new audience in the planetarium. We hope it will connect visitors to regional and global importance of their local institutions as they learn more about life on the planet. This innovation creates endless opportunities for new public programming to be developed, can be used for behind-the-scenes tours, and even could be used to host research seminars. Just a few weeks ago, I attended my very first planetarium conference to teach a workshop on BioViz. The tools were very well received, and there was a lot of interest in the Digistar user group to connect with more scientists. We discussed hosting a workshop to connect the collections community and planetarium educators that would be aimed at developing new mutually beneficial collaborations between unlikely partners. I've absolutely loved the opportunity to take over the dome, which, by the way, you can drive with an Xbox controller. And I'm inspired now. I'm dreaming of all the endless possibilities of communicating the importance of natural history collections and the science that we enable. I encourage you to reach out to your local planetarium to see if they run Digistar, or connect with me if you'd also like to be involved. This project was supported by funding from the National Science Foundation, and I'd like to acknowledge the fantastic teams at Evans and Sutherland's who turned my dream of digitized data in the dome into a reality. Also, the planetarium staff at the Cleveland Museum of Natural History have been incredibly welcoming and encouraging throughout this entire project. And I'd also like to thank the organizers of this symposium. Thank you.
Thank you, Nicole. Everyone is just very excited about all the things that you're doing and how you're bringing collections to a completely new tool that we can use to engage with the public. That's really, truly amazing.
I have to say that the first time I ever saw my dung beetles projected on the dome, it was super magical just because of how immersive it is. And I really, really hope that everybody gets to see their own stuff in a planetarium one day.
Nice. I have a question. How much, I mean, the only experience that I have had in a planetarium is just like you have, it's like going to the movies where you enter the room and there is a story prepared, right? How much preparation does it take to make one of these stories to tell people?
So it really depends. Like it is, you know, there are many of the planetarium shows are entirely relatively scripted, but there's always a lot more engagement and audience interaction. You can take it in any sort of direction. So the best thing is about this, the software that we use is you can navigate the globe and the earth as you talk about whatever story you're trying to tell. So what we're hoping for the program that I will be eventually developing is that it will integrate these sort of maps as well as documentary style footage. So we have a spherical camera. You know, I love dots on maps, but I feel like most of the audience probably wouldn't be so into it. So you've got to mix it up a little bit. But there is the element of, you know, having some of the stories and some of the data sets prepared in advance. But the great thing is about the IDBio tool, if someone asks me about an entirely different species that I've never thought about it, we can just quickly search it in the portal and hope that there are enough records to be able to answer the question.
Okay, good. We have a question from Nicole Fisher. I think you said there is 167 planetarium. There was a map of them, and how do you find where they are? I think there was like 4 in Australia.
Yes, so the map, so there are at least 165 planetariums that run Digistar 6 and 7. Of course, this number always changes. There is a World Planetarium database that tells you which planetarium wherever runs what software. I know that within Australia, the planetarium in both Brisbane and Victoria run Digistar. So the first thing I would do is check the World Planetarium database for what software that they run. Alternatively, if you just reach out to your local planetarium, they might be able to tell you as well. But yeah, I'm super excited about the potential reach, but at the same time, you know, unless there is interest from both our collections communities, as well as the planetarium educators, it may not ever be adopted. So I'm really trying to connect the two communities and hope that one day biology data is just as common in a planetarium as looking at the stars.
I guess my next question is how much time, like, I don't know, like thinking of this as a sort of specific tool from the collections perspective to take this into a new venue, like my guess is that you would need a person that has both components in their head to be able to bring this to happen.
Yes, but you know, the educators that are front of house at planetariums, they all have very diverse backgrounds. Some of them are definitely into astronomy and other things. You know, many planetariums these days do teach, you know, components of evolution and other stuff as well. But I feel the hardest part is identifying the right story that, you know, the data that exists is really visually compelling. And, you know, assisting the planetarium educators to work out, like, you know, how do I get data from an aggregator? What's the best way to filter it? What is, you know, when is, you know, there are always dots in the data that need to be removed. And the good thing with the fact that they're KMLs, you can open the data sets in Google Earth and remove any erroneous data points and then resave it. There's just so much potential, and I really do want to connect people. I know that I can see many people in this audience that I know that they have local planetariums that run the right software. So, you know, hoping to do a workshop with the two communities maybe very soon.
All right. Well, we don't seem to have any more questions from the audience. So I guess, thank you so much, Nicole. That was really, really interesting and really, really inspiring. Like, I think everyone now is gonna check out the closest planetarium to their location. All right, we are up for a break right now. We will be back in 14 minutes, 13 minutes.
I think, yeah, 13 minutes, sorry.
All right, I'll see you in a bit.
All right, so we're gonna go ahead and get started. We'll begin this second block of From the Cabinet to the People with Isabel Betancourt from the Academy of Natural Sciences at Drexel University with in-person virtual partnership and through the news outlets. An overview of how the entomology department at the Academy of Natural Sciences of Drexel University makes its collection accessible beyond scientific research.
Hi everyone, my name is Isa Betancourt and I'm a curatorial assistant of entomology. About to give you a tour of how the entomology department at the Academy of Natural Sciences of Drexel University makes its collections accessible beyond scientific research. For context, it'll be helpful for you to know that our collection is part of a 209-year-old museum in the heart of Philadelphia, Pennsylvania. We welcomed over 204 visitors into our public museum in 2019, and over 22,000 school kids visited the museum that year too. Behind the scenes of the public museum are thriving research labs and operations. There's the Patrick Center for Environmental Research that focuses on studying and protecting watersheds. And there is the Center for Systematic Biology and Evolution, which curates and studies the 18 million natural history specimens whose collection dates span the 209 years of the museum's existence and even go beyond it. Within the 18 million natural history specimens are 4 million insect specimens. They are protected from disturbance and light behind the scenes in these metal cabinets. The entomology collection is accessible to our team of curatorial staff, student workers, and volunteers who work with the collection and study it, along with occasional long-term visiting researchers. Like a typical functioning institutional research collection, the collection is also readily open to visiting researchers from around the globe. As I think all here can agree, the entomology collection is a priceless resource that is full of natural history stories and information. Unfortunately, we cannot simply open the collection space for the public to freely explore on their own. Access must be planned and guided to protect the specimens and optimize experiences. We connect the collection with the public in ways that can be roughly categorized into 4 categories: in-person tours and events, conversations with news outlet reporters, event partnerships with outside organizations, and virtual media offerings. We'll touch upon each of these 4 items across the course of the presentation. I'm heavily focusing on in-person events in this presentation, and as a heads up, I placed 2 communication concepts in with examples to offer new ways to examine and arrange outreach events. Here we go. We have 2 annual events at our museum where we take research specimens and our entomology staff out to the public museum space for viewing and interpretation. They are Bugfest and Members Night. Bugfest welcomes over 2,000 visitors into the museum. There are live bugs, games, and activities across the entirety of the museum. A whole hall of tables are put together and staffed by entomology curatorial staff, students, and volunteers. We have insect specimen drawers on display, as well as insect specimens in preparation, as you can see in this picture with the spreading boards. We also typically display live local insects we've captured or that we are rearing. I love having the dissection scopes available for people to try at these events. Visitors have a lot of fun using them and experiencing the crisp view. One time a kid looked at a live assassin bug that was upside down on the lid of a petri dish and exclaimed, "It felt like it was on my eyeball." Members Night is a special night for thanking our members for their interest and support. This year it was in the main museum because of COVID, but usually museum members can explore the behind-the-scenes spaces where the staff and scientists set up stations with activities and displays showcasing parts of the collection or their research. One year for Members Night, because it was close to Halloween, we chose the theme, The Great Insect Pumpkin Patch. We assembled an array of orange and orangish insects. We had a variety of insect orders represented in the patch. It may seem like a simple theme. I thought it might be too simple at first, but the color theme turned out to be a great foundation for creating the display. Overall, the theme was very relatable and sparked imaginations. We decorated the table with gourds and Halloween lights. It was simple but allowed space for creativity and dives into sub-themes. We invited guests to explore coloration and its functions. Members vote for their favorite stations, and to our surprise and delight, the Great Insect Pumpkin Patch won honorable mention that year. This Members Night display used the element of surprise to get people's attention. Most people think of moths and butterflies as bright and beautiful, and perhaps even simple. This exhibit took words associated with Halloween fright and highlighted an example in the Lepidoptera world that exemplified each. The examples explored a mix of science and folklore. In addition to the annual events, there will occasionally be one-time events that pop up like Door 19 and collection tours. I categorize tours as unique events because we don't have a regular tour schedule. It happens upon appointment. Small groups of around 10 people or less work best in the tight collection space. We've given tours to local school groups, especially high school and college groups, to politicians, museum donors, artists, family, friends, colleagues, local community members, etc. And we tailored the tour as much as possible to the groups. A huge benefit of these in-person events is that we are largely meeting people one-on-one and can cater to their interest level, attitude toward insects, and their knowledge level. The social judgment theory is a communication theory that explains the importance of understanding the audience and meeting them where they are. Long story short, if we don't meet them where they are, we risk pushing them the opposite way. Okay, back to the events. Door 19 was a quarterly themed adult ticketed after hours event with live music, collection tours, local food, and activity stations. Entomology was invited to participate during the Speakeasy or 1920s Prohibition themed edition in 2018. We set up a ladybug ID station. We set out unidentified ladybug scopes and online and printed resources for identifying ladybugs. Guests came by and we helped them go through the ID process. Once they came upon an ID, we'd help them write their names on the ID label and they'd put it on the specimen to be forever preserved in the entomology collection. Artists occasionally visit the collection to study specimens for their work. This is Mary Capaldi, who visited our collection many times to draw the moths and beetles. This brings us to exhibits, which I will categorize as unique events extended over a period of time. The pinned insect art and insect science exhibit lived in the Art of Science Gallery at the museum. It featured the artwork of Christopher Marley on one side of the room, which you can see on the left of this slide, and the entomology specimen preparation process, which is not quite as glamorous, on the other. I'd like to introduce another communication approach here for us to contemplate when putting together or evaluating outreach. It is the elaboration likelihood model. It says that people process messages in two main ways. They may use surface level characteristics, or they may think critically about the rhetoric of the message. Often the approach will be mixed. Time, jargon, or ease of understanding, a person's enjoyment of thinking, and finally, how relevant the message seems are all examples of factors that play into the route a person takes to evaluate a message. If conditions are conducive to a person taking the central route rather than the peripheral, the message being shared will have a greater impact and hold, if the rhetoric is sound. Now, for an example, a captive audience has more potential to be in a condition of high elaboration because they have set aside time for the message, while a non-captive audience at a festival may be less able to elaborate and take the central route because of the potential distractions and frenzy of such events. When it comes to pinned, visitors will see the beautifully designed pinned insect arrangements by the artist. The heuristics are likely to influence a positive impression about the insects. The appealing aesthetic design may be a gateway for drawing attention, sparking interest, and then raising the elaboration likelihood of the guests so that they explore and dive more into the content of the exhibit. The Clergy and the Academy's collection was a really great example of an exhibit pairing with local events. The Pope was visiting Philly, that my coworker Greg Cowper will discuss in the presentation following this one. This exhibit, Bugs Outside the Box, featured larger-than-life, scientifically accurate insect sculptures by artist Lorenzo Pesenti. It was on its way to the museum when our department was about to go through a major cabinet renovation. Our open-shelved storage units were being replaced with modern metal cabinets with doors. As a collection in a 200-year-old building in a large city, space is always a concern. We coordinated with the exhibits department to have the exhibit built in a way where we could have space to temporarily store some of the entomology collection during this renovation. You can see some of the storage cabinets and drawers in the background of the middle photo. The collaboration also resulted in the construction of a box for entomologists. You can see entomologists inside the box at work. Visitors were able to walk up and interact with us. Like the entomologist box, there's an opportunity to host this sort of live working presence at this Science Live space in the museum today. Here's an exhibit that was up for a full year. It was the museum's bicentennial exhibit. It celebrated 200 years since the Academy's founding in 1812. This exhibit was up for a full year, and we didn't want to risk damaging research specimens through light exposure, so we bought expendable specimens that had a high visual impact for our department's contribution to the exhibit. And that's the end of this section. But before we continue, I want to share a few notes about the in-person events. So we transfer insects for display into Academy drawers with special UV glass to help protect them while they are on display. A nice side effect to curating specimens for display is that the insects selected and their drawers end up receiving curatorial attention. That's a big deal for such an old collection where many specimens are still in cork and not yet in unit trays. Also, I think it might be helpful to share that we do not get extra funding for these events. We work with what we have. However, I ought to point out that as a part of a public museum, we sometimes have the benefit of collaborating with the exhibits department and videographer. On to the news. When insects arise in the news, we are often called for comment. Topics have ranged from mosquitoes to orb-weaving spiders to spotted lanternflies to ants. One summer, swarming insects that were getting in the faces of Philadelphians made the news. The news channels didn't know what they were and even were calling them gnats. They came to us and we were able to confirm that they were ants. We happened to have collected some earlier that day. The record keeping that occurs through the entomology collection allows us to answer questions about natural phenomena that we experience as a society, like that of the periodical cicadas, which emerge every 17 years. We shared evidence-based observations and hypotheses, and we provided cicada specimen visuals to news outlets this past May 2021 during the emergence. Working with the news is unique because they are a rushed outside organization looking to fit you into the story they are reporting on. In my experience, they don't tend to share their agenda or story plan unless you ask, so be sure to ask. That brings us to partnerships. Partnering with an outside organization has been a great way to reach new communities of people. We have partnered with Atlas Obscura to provide virtual tours of the collection and also an on-site entomology workshop. We've also partnered with Science on Tap to present in their series of weekly sciencey presentations at a local Philadelphia bar. A super cool partnership comes from our next speaker. Academy curatorial assistant Greg Cowper was selected to pursue his proposed art installation at the Eastern State Penitentiary. His project, Specimen, was inspired by an 1890 publication of entomological news featuring a list of insects collected by an inmate at the penitentiary. For his installation, Greg collected insects and crafted a stunning gallery to display the specimen findings within the ESP's own museum. Greg's partnership with ESP, established in 2011, continues to be renewed. With that, we've come to the fourth route, and likely one that we all have become decently familiar with because of COVID: virtual events. Here's a list of some of the virtual events we've done to connect our cabinets with the people. I'll touch upon 3 of them. The Bugscope Live broadcast. I started live streaming from the collection in 2017 to give this feature, which was new to social media at the time, a go. The weekly broadcasts have connected with viewers both in the States and around the globe. Live streaming can be like an online version of in-person tabling because people can come and go and ask questions that I can immediately address, allowing for that personalization. I presented about this project at ECN in 2017. I'm happy to return and share that this project was selected as runner-up for the Entomological Society of America's 2021 Science Communication Award. Live broadcasting is exciting and offers lots of creative value to the audience. However, it's not always evergreen or polished and efficient with a high replay value. Our department has put out several polished videos made in coordination with the museum's videographer that have had great success on Facebook, such as this one about light trapping, which has 35,000 views. With the arrival of COVID, our department came together to create 3 butterfly guides that featured specimen photos taken for the LEPNet North American Digitization Project to train an artificial intelligent ID program. It was a wonderful use of the photographs for general purpose that anybody in the region can use. One guide featured early spring butterflies, another late spring, and the 3rd listed summer butterflies. They were published on the museum's blog, and the graphic designer at the Academy even worked with us to put together a printable version. This concludes our time on the tour. As a concluding thought, I'll say that this is an exciting creative challenge. It's an exciting creative challenge to craft an event or display to connect specimens with the public, and it can be very surprising and rewarding to see how programs are received and interacted with by the audience. So thank you for coming on this tour of our collection's activities beyond scientific research. I hope you found it interesting and helpful.
Thank you so much, Isa, for your presentation. I really loved seeing all of the in-person events that you guys are doing with your collections. Are there any questions from the audience?
Yeah, I do realize that I went through that very fast. But I had so much I wanted to share with you all and wanted to give a tour and overview and share ideas. So yeah.
They were great ideas. I have tons of ideas now from all of this. So I think everyone's really excited about it seems too.
Yeah.
Oh, Floyd, how do you manage to find time to do anything else?
Yeah, so I didn't really give a sense of the timeline of when all these things happen, but these are events that have happened across the last 10 years. About the bicentennial was 9 years ago now, and then the pinned exhibit was in 2013, and so it's all spread around. But yeah, it definitely takes up time, and but it's something that I know that I'm super drawn to. Science communication, yeah. So,
Isa, just out of curiosity, who are the members? You talked about members night, but it's
sure.
like a membership for the museum or something like that?
Yeah, so if you think of maybe your local aquarium or science museum or art museum, It's sort of like the general membership where you pay like a certain amount of money. Usually it's like $100 or something. You can go as many times as you want throughout the year and bring your family. That's what it is. Yeah. So that was a museum-wide event, not just entomology. Yeah, to be clear too.
All right.
We have a few questions in the chat. Can you talk more about the Ladybug ID event that you guys did? What key did you use that was public friendly?
Sure. Yeah, so we use ladybugs that are specifically like the ones that look like typical ladybugs, if you know what I mean, for this event. Red and black dots that were collected in Center City, Philadelphia. And we used, I'll have to go back and look, but there is a guide online that's a clickable guide where you can click and you're not stuck with just one dichotomy. I can get that information for you if you send me an email. Yeah.
Okay, great. And we have to move on, but if anyone has any questions, they can reach out to you. And we also have a meet and greet afterwards as well, too. So thank you so much again. So next up, we have Greg Cowper from the Department of Entomology, also the Academy of Natural Sciences of Drexel University, with this talk, A Tribute to Pope Francis, Specimens from 200 Years of Natural History Collections.
Hi, everyone. Welcome. I'm Greg Cowper from the Academy of Natural Sciences, Department of Entomology in Philadelphia. In the late summer of 2015, we got word that Pope Francis was going to visit the United States and Philadelphia in particular. And up to 2 million people were kind of guesstimated to maybe be on hand for his visit. So we at the Academy thought, how do we get people into the museum? How do we bring the cabinet to the people during the Pope's visit? So we came up with the idea of an exhibit called The Clergy and the Academy's Collections, which simply was to highlight people of faith's contributions to our many collections at the Academy. So lo and behold, the Pope shows up. It's September the 26th, 2015. He's coming down the Parkway in the Pope-mobile. My wife and I were there for the festivities. The Parkway is very close to the Academy, by the way. But the first thing we had to do was come up with a poster for the exhibit, which we had done prior to his arrival, of course. And this is adapted from The Master of the Bardi, St. Francis and Scenes from His Life. It's a 13th century artwork. And I just substituted all the scenes of St. Francis's life with material that had been donated by clergy to our collection. So next we had to come up with what is the relationship the Academy actually has with religion. So we turned to our founding document here, March the 17th, 1812. And it says, number 2, in order to be a member of the Academy, it's determined to be a member of the Academy, you must have a mind totally free from all religious and political prejudices, preventions, and animosities. And if we delve farther into this, and the transcription will help, or which shall be uniformly capable of divesting itself in this society and in all things in which its affairs or interests are concerned, perfectly from all preventions, prejudice, animosities, habits, and expressions appertaining to political or religious differences or distinctions, dogma, or argumentation. So basically what this means is, sure, religion, sure, politics, but just leave them at the door when you come to the Academy. So another thing we had to do is we queried the collections as to, do they have specimens from clergy members? And also our archivist, Jen Vest, shown here, sorted through all the membership cards, 200 years of membership cards, to find out who clergy members were that might have been members of the Academy. And in this vitrine here, we displayed over 60 of them. And examples are the Reverend Wayland Hoyt, James M. Niblo, also a Reverend, Sister M. Martha, and Rabbi Max D. Klein. The first person that we highlighted in the exhibit was the Reverend Frederick Valentine Melsheimer, who was a Lutheran clergyman. Melsheimer was famous for publishing Catalog of Insects of Pennsylvania, which was the first separate insect publication in America. And I draw your attention to underneath his name, it says Minister of the Gospel. 1,363 species were listed in Reverend Melsheimer's catalog, including the giant stag beetle here on the left from the northeastern United States. And also from the northeastern United States, we have Dynastes titius, the eastern Hercules beetle. And I've been doing this since I was a kid, insects in cigar boxes. So when I could, I stuck a couple of them into the exhibit also. We're next going to talk about Reverend William Jacob Holland. He was a Presbyterian minister, and he was director of the Carnegie Museum. An example of a vitrine in the exhibit is his vitrine. You can see the materials on display. But what Holland was important for is he corresponded with our department head at the time, Henry Skinner, and borrowed specimens to complete missing illustrations in his books. And his books, I'm referring to his butterfly book and his moth book, all in the late 1800s, and the moth book was 1903. So we see here on the right an erebid moth, which Skinner provided Holland to illustrate for his moth book. And on the left is the Parnassius butterfly, which wasn't from our collection, and it's from our collection, but it wasn't illustrated. It's just shown in his book. Next, we got Reverend Dr. John Bachman. He was pastor of St. John's Lutheran Church, Charleston, South Carolina. Bachman was really prodigious, and in my view, his claim to fame was not only was he a naturalist, but he was a friend of John James Audubon, and the two of them corresponded and worked with each other. Here we see Bachman's warbler named in his honor. Now it's probably extinct. It was recorded by Bachman in 1832, and the specimen here in the box is from our ornithology collection, and it was collected by Bachman. Here we have the pronghorn skull, Bachman's warbler again, Bachman's silhouette, and here's the viparous quadrupeds of North America, which of course was Audubon's tome after Birds of America. He did the mammals, and Bachman wrote the species descriptions and the text for this celebrated work. The red-bellied squirrel is shown here, which he provided Audubon to illustrate, and also Audubon painted Lepus aquaticus Bachman, which Bachman described this rabbit, and it ended up in the book also. Here we see Heinrich Muhlenberg. He was a Lutheran minister, pastor of Holy Trinity Church in Lancaster, PA, painting by Charles Wilson Peale. He was noted as the father of American botany. Several of his works on botany of the eastern United States were the first of their kind. He also discovered and identified the bog turtle, Clemys muhlenbergi, which was named in his honor. Here's the bog turtle from our herpetology collection. He was mainly noted as a lover of grasses, and he had a genus of Muhlenbergia. It's called Muhlenbergia, was named in his honor, and another herbarium sheet from the Academy's collection. In the exhibit, I displayed some herbarium sheets of Muhlenberg's across from the encyclical of Pope Francis, and it just fit exactly, and a reading of it shows how closely it's aligned with the strategic plan and mission of the Academy. On care of our common home is this portion of the encyclical Loss of Biodiversity. Greater investment needs to be made in research aimed at understanding more fully the functioning of ecosystems and adequately analyzing the different variables associated with any significant modification of the environment. Because all creatures are connected, each must be cherished with love and respect, for all of us as living creatures are dependent on one another. So it's a beautiful piece, and the encyclical was quite inspiring to readers of it. Next up is the Reverend Henry McCook, who became a favorite of mine. He was pastor of the Tabernacle Presbyterian Church here in Philadelphia. But one claim to fame was he was vice president of the Academy from 1882 to 1900, and also president of the American Entomological Society for 2 years. But McCook was an accomplished artist. Here's a watercolor of Reverend McCook's: an underground dairy, ants tending aphids feeding on root. He also described the basilica spider, Hentria basilica, and you can see it gets the basilica name from its commonality with the dome of a basilica. And lo and behold, directly across the street from the Academy is the Cathedral Basilica of St. Peter and Paul, where the Pope also spoke. And I nosed around over there, and as circumstances would have it, there was this art exhibit which was very spider-like in its showing, and I just thought that was neat because of McCook's basilica spider. Here's a drawing he did of the mandibles of the Texas cutting ant. Moravian Bishop von Schweinitz, who was a botanist and named more than 1,000 new species of fungi in the early 19th century, an endeavor which earned him the title of Father of American Mycology. Here's a couple of his hand-colored illustrations. Next up, Joseph Clemens, Reverend Joseph Clemens, and his wife Mary Knapp Strong Clemens. He was a missionary Methodist minister, army chaplain, naturalist, and botanist, and he donated to the collection the reticulated pufferfish, which when we got all the fish together that clergy had donated, there was about 16 jars in this Victorian vitrine on display, and Clemens pufferfish is down there at the bottom in the middle. Niceforo Maria, brother Niceforo Maria, really was one of the rock stars of the exhibit. Just his birds, look at all the birds he donated to the Academy that we found in ornithology. The material he sent to researchers led to the description of more than 130 new species of plants, arachnids, crustaceans, fishes, reptiles, amphibians, birds, and fossils. Quite a prestigious endeavor. So Brother Niceforo, again, here he's the wire-tailed mannequin that he collected from the, showing his labels from the ornithology collection. He collected this anole, South American land snail. And next up, we move on to Africa with Alden Grout, American missionary known for his missionary activities in Zululand, the northeastern section of present-day KwaZulu-Natal. And he donated the thick-tailed galago, commonly known as the bush baby. You can see his handwritten label, Zulu Mission, South Africa, S.A. Grout, there in the left-hand corner. Another African minister, Reverend Robert Hamill Nassau, worked in Africa for over 40 years. He gave us this gorilla skull, which was collected in Gabon. So near the end of the exhibit, I had collected memorabilia about the Pope's visit, and I was able to get it displayed in the exhibit before it ended. And here's 2 vitrines of Pope Francis memorabilia collected in Philadelphia during the Pope's visit. And who would have guessed that only in Philadelphia could the Pope be honored by putting his likeness on a pizza box? So thank you to everyone for attending the webinar, and of course to the many individuals of faith that have enriched the Academy of Natural Sciences collection over its 209-year history. Thanks very much for attending, and hope to see you again.
Thank you so much, Greg. That was great. I had a question. You might have answered it, but did the Pope ever get to see the exhibit, or was it ever gotten? Do you know if it was ever like shared with him to have gotten to see?
The Pope was a no-show.
Yeah.
But that's a good question. I don't know if it was ever shared with him, but you know, now that we've got this recording, we can just send it to the Vatican and
definitely
see it there. The attendance kind of was underwhelming, to tell you the truth, because I think I mentioned in the beginning that up to 2 million people were expected. Really only 1 million people really showed up in Philly for the event. And I mean, it was tight in terms of, you know, making sure the Pope was safe. And the Academy was actually closed for many of the days that the Pope was in town, just because of like he was speaking at the Basilica or doing events near the Academy. And they built barriers, but we were open a couple nights late, and they asked me to stay there and interpret. And we did get a lot of people that had come for the it was the gathering of families in Philadelphia, and many of them did come in the evening to view the exhibit and, you know, discuss natural history. So it was pretty cool in that sense.
Yeah, that's great. That's awesome. Seeing if there's any questions.
I think one of the great things about it is all the historical component, just bringing all those specimens and all those stories just like digged out from the, I guess, museum documents. How long did it take to prepare the whole exhibit?
It was about a month to probably 6 weeks of getting specimens from the different ologies, so to speak, at the Academy. I think there was eight different collections plus the library and archives. And then literally it was a 2-day, 48-hour marathon to set the whole thing up. So when it opened up, I had literally been awake for 2 days. And but that's another thing that made it really fun, actually. And then people's reactions that did come, it was pretty cool. And, you know, kind of mirroring some of the things that Issa said. A lot of those things got recurated because of the exhibit. Things were broken, put back together by the preparator in vertebrate paleontology, Fred Mullison. A lot of things got moved into UV cases. A lot of nomenclature was changed. I mean, you know, 100-year-old nomenclature is not up to date normally. So some things did get changed. But anyway, it was a pretty cool experience, and just finding how much material we really do have at the Academy behind the scenes. You know, only a small bit, as we all know, of the collections are really on view to the public. So it was pretty cool in that way too.
definitely. Thank you so much again for your presentation. But we have to move on to the last speaker of today, which is Susan Wright from Entomology at the Queensland Museum, presenting about insects on display at the Queensland Museum, past, present, and future.
Hi, everyone. So I'm one of the collection managers at the Queensland Museum. We look after about 4 million specimens. And so I'll talk today about some of those specimens that we've had on display, both in the past and today, and hopefully give you a little bit of a rundown about what we might be doing in the future. Okay, so this was one of our old buildings. So we moved in there in 1899. And this is what the gallery's floors looked like in 1930s. So lots of old glass cases there. It was a lot of natural light. They didn't have electric light until 1943. There's a lovely little story about a duke who had a private tour there, but because there was no electric lighting, in order to see any of the exhibits, they had to light a match for him. So most of the exhibits were put together in the 1920s, and they really didn't change until we moved to our new building in 1986. So they were very static, but these are some of the specimens that I found in the collection that were taken out of those displays in the 1980s. So you've got the handwritten labels by Henry Hacker, who was a curator at the museum and put together a lot of those entomological displays, and you can see these beautiful Hercules moths here. Now they're in pretty good condition considering they would have been on display for nearly 60 years, and the reason for that is that the Ento collection actually put cloth over those cases so that you actually had to lift the cloth up in order to see what was under it, and that did protect them against a lot of that fading, which is so prevalent with these sort of coloured specimens that have the pigments, so browns and reds and yellows, etc. So they're looking really, really good nick, considering how old they are. So 86, we moved to our new building, and we of course had insects on display there, but the major exhibition we put on was in 1992. So this was a major collaboration between all of the entomological institutions in Brisbane, and it went on for 4 weeks, and we had about 75,000 people come through. And the main aim of that exhibition was to basically promote entomology and entomologists, and just to get people to realise a little bit more about some of the insects in their backyard, and to reawaken, I guess, that awe and wonder. I'm not going to show too many slides about that, but this is one particularly nice display. So this took the Australian National Insect Collection, the Australian Museum, a lot of private collectors, the Queensland Department of Primary Industries Collection, and the Queensland Museum Collection, in order to gather all of the Australian butterfly species in the one place at that time. So we've actually got a few more species since then, but the way these are displayed is this is actually a cutout panel, and behind that is insect drawers, so metal insect drawers. The insects are pinned in the bottom, and then the drawers are stuck up vertically, and the panel is stuck on over the top of that. And you can see the graphics is very different to what we would do today. So moving on to a much more modern exhibition. So this is Museum Zoo. It was put together with an external design company, Brandy, and it was in 2010. So it was up for a few years. So it was considered to be a more permanent exhibition. It's been down a long time now, but basically it was based on size. So animals size and me. So it was a parade of taxidermied animals and insect and animal specimens, going from the very large down to the very small. So we've got the spiders down here, and then of course the insects are right at the bottom. So we had our own cases, and in order to be able to see and get up close to those specimens a little bit more, they had these magnifiers on railings. So the exhibition spoke about why insects or why animals were big, why some were small, why weren't some as big as they used to be. So for example, the big dragonflies that used to be so big, why don't we get really big dragonflies today? A little bit about exoskeletons and things like that. And then of course the big panel at the back here is a lot about the megafauna and why maybe they died out. So this is one of our cases. We still actually have this case today. It seemed too beautiful to dismantle, and it's got a Perspex case on it, so it's perfect for us to take out and show people without having to worry about that glass. So you can see there's some little teleoids and ceoids and things down the bottom here. So people would have needed that magnification in order to be able to see that there is actually something on that little card. So moving through the galleries today, level 4 is basically where our entomological collections are concentrated or displayed. This is our section in sort of the taxonomic area, if you like. So we've got more species in there than any other group did, obviously. And we've really concentrated on the insects that are in Southeast Queensland. So really trying to concentrate on those specimens that people might be able to be recognising and trying to identify. So when they come in, they might recognise something there. These specimens are all pinned onto polyethylene foam. So because this is a permanent exhibition, we'll probably need to swap out a lot of those specimens. They might fade, they might break. The foam is quite good and it absorbs a lot of vibration. So as people or kids run around or thunder around in these areas, some of those cases will vibrate, and certainly some of the, for example, freeze-dried spiders, that certainly damages them. But these insects, because we've managed to get them into that foam, that helps protect them a little bit. And it's really easy to open these cases, reach in and swap those specimens over. So we also have these drawers here. So while the entomology section really put the specimens together for this, the Discovery Centre people would be more likely to work on these drawers. So there's another image here. So they've got various different depths so that they can put, say, insect nests or things in it. This is our lot here. And so this is a jousting beetle section. So again, there's foam down the bottom so they can pin insects in and they can swap the text over, so that if there's something particularly interesting or happening that people are talking about, maybe there's a butterfly migration or something like that, they might be able to put some of those specimens in response to some of those major inquiries. So moving along next door, there's the Wild State, which is another one of our permanent exhibitions. This is much more visual. There's a lot more colour. It's not as, I guess, sterile as that area and clean as the Wild State looks. So we've got projections on the ground. There's audio. We've got images around behind all of the objects and specimens. So this is a story about Queensland's unique animals and their habitats. So this is the rainforest area that Chris Burwell here is our senior curator. He helped develop this particular section. And so he's put a whole heap of rainforest insects there. And the idea was to get as many of the species out of the cabinets and into this area as possible, so people would really start to appreciate how varied our fauna was in Queensland. And so we don't actually have a lot of these species identified in the labelling, which means, again, because it's a permanent exhibition, we'll need to change them out. We can do that a lot more easily without having to change labels and things like that. And again, they're on polyethylene foam, which is really nice and secure. They're not going to fall, and we can move things a lot easier. Okay, so those are our permanent exhibitions. So we also have things that we wouldn't put on permanent exhibition. It's just too risky. And generally we assume that anything that goes on display down on our floors for any length of time is probably going to get damaged. So we wouldn't be putting something that's really valuable. We wouldn't be putting types, and we generally don't put registered specimens down there either. We might swap a few of them over if they start looking particularly bad, but we'd like to be able to leave them down there for as long as possible and save, say, the rest of the specimens that are more scientifically valuable. So it's duplicates and things like that that we would tend to put down there. But all of those specimens that maybe are more scientifically valuable or older, for example, or more fragile, we can bring them out in temporary times like these. So these are specific events where we would be really interacting with the public, interpreting what they're seeing, answering their questions, and they're really valuable for both the punters and for us. It's really rewarding for us to get that feedback from people about how excited and how much they're learning about insects when we actually get down there and talk to people about it. So going around the circle, this is World Science Festival. So we had about 75,000 people come through in a week in that particular event. And this is Karen Kosh, the other collection manager here, and Vivian, one of our honoraries. And Karen's actually got a drawer there of insects that belongs to a collection that's well over 100 years old. So that's not the sort of thing we would generally bring down on the public floors, but people really appreciate the effort that we go to bring these things to them and explain to them what it is. So it's basically just a big drawer of lots of golden scarab beetles, which is really impressive. It catches people's attention. They come over and then we can have a bit of a discussion about specimens and how long they last, or the species, or whatever else they want to talk about in response to what we've got on to show them. Meet the Curator is down in the Discovery Centre. That runs every Wednesday. That's a rolling series of talks from all of the curators and collection managers in the museum. So we're a cultural institution as well as natural history specimens. So, you know, there's all sorts of things can be down there on a Wednesday. And that's a really nice, intimate little group. You don't tend to talk to too many people unless there's school groups going through. Halloween can be a bit like World Science Festival, just take down a whole heap of things. It's family orientated. So you can see there, we tend to carry things down there that kids might be able to touch. So lots of handling of specimens and things. Alcohol specimens that have been partially dried out are quite good for that, we've discovered. And this particular one, so the other three are within the museum, on the museum gallery floors. This one here is taking the museum out to the streets. So essentially they take a couple of trolleys with a whole heap of insect and other objects from the museum. It can be cultural, can be biodiversity, and they'll take them over into the Queen Street Mall, which is over the bridge, the central part of the Brisbane city itself, and just basically talk to anybody who comes up. And so they've had some really good response with that, but people just saying, I didn't even realise that the museum had these sort of things there, and I'll have to come and visit the museum one day. So they've had some really good feedback there and quite enjoy doing that. So that's a pin specimens. That's what we do with them. So a lot of the look of what we've had with the pin specimens is in that foam. We don't have a lot of diorama material at the moment. Sometimes we do, but we don't have to have any of that on display a lot at the moment. So the imagery these days is really important. So in that Discovery Centre, we used to have much more really tiny specimens to try and show people that insects were really small as well. But feedback from Discovery Centre basically said that, look, it's not worth it. People either think that the specimens have fallen off or been eaten or something, or they just can't get close enough to see it and they don't really get that idea. It doesn't work. It doesn't bring that idea across. So what we do these days is we've taken most of the small stuff off display and we tend to use imagery instead. So instead of showing them the small things, we make them bigger. So this particular photo here, you've got a narrated, it's an auto montage image. So on each end of every one of these Discovery Centre cases, there's a slideshow. It's not only showing insects, that's what I've shown here, but it also shows all of the cultural and other biodiversity objects, but it's all behind the scenes collection items from the museum, and that works really, really well. So the graphic designers go a little bit crazy with insects somehow. They stick them on bridges, and they also stick them up on the hoarding. So these were pretty like life-size insects. This is a Pelorodiad, which is really tiny, and so we've managed to blow that up really quite large. And people would stand in front of this butterfly here and take photos and put that up on social media. So it was quite a good little idea there, instead of having a great big white wall, which is what we had before the insects went up. So we've had a few failures. They did try to put some stickers up on the outside of the glass on the museum. So this used to be our logo, the Queensland Museum's logo, the birdwing butterfly. But unfortunately, because it was on the outside, the temperature changes meant that sticker started contracting and expanding, and they were concerned about fractures in the glass. So unfortunately, we won't be able to do that anymore. So I mentioned we had an exhibition coming up. So in 2023, the Entomological Society of Queensland will celebrate their 100-year anniversary. It's the oldest entomological society in Australia, and based here in Brisbane. And so again, much as Friends and Foes was, we'd like to engage with all of the other entomological institutions in Queensland and put on an exhibition just to promote insects, entomology and entomologists. And I know these aren't insects, but these are some of the other sort of marketing things that we would like to be able to do for the insect exhibition as well. So this is an inflatable peacock spider. So this was developed in conjunction with the Spiders exhibition, which came from the Australian Museum and came to the Queensland Museum last year. Unfortunately, it had to be closed because of COVID. But this guy here is Webster, our puppet, and you can see the feet of the puppeteer there. So he actually gave behind the scenes tours through the museum during COVID. So if you want to have a look at those videos, they're really cute. He's got little palps that move and all sorts of things. Just look up Webster in Queensland Museum and you should be able to find some lovely videos of him. So we'd love to have a puppet insect and maybe a big inflatable on the building as well. And just lastly, this is a drawer. It's 100 years old. So this is part of a collection that toured from Kuranda to Adelaide, so 3,000 to 4,000 kilometres in 1918. So one of our earliest travelling exhibitions. And I'll just read that out. And whenever the way seemed long or his heart began to fail, she would sing a more wonderful song or tell a more marvellous tale. And that was written by Longfellow. So thank you very much. I hope you've enjoyed my tale. If anybody would like to get in touch and have any more questions, then don't hesitate. Thank you.
Thank you so much for your presentation, Susan. That was really great. I loved seeing all those different types of exhibits that you guys were able to put on. Are there any questions?
I think now everyone wants to visit your museum. It looks really amazing. The exhibits are just like, I don't know, it looks like well lit, and I don't know, the light makes all the cases and everything like you want to be there, definitely.
Oh, that's good feedback. Yeah, there's so many things I could have put on in the talk, but with 15 minutes, I sort of tried to pick some of the things that maybe people hadn't seen before, like the puppet and the big inflatable on the roof. They were a lot of fun.
Yeah. What do you think is like, what are the take homes that people can, I don't know, I guess, do you have any feedback from people? Like, do they like some exhibits more than others?
It depends on who you've got coming through. A lot of the people who come through are obviously families with small children. So of course they want a take home. They want to do an interactive. The kids want to do some colouring in, so they want to colour a beetle or they want to make a mask or something like that. And so a lot of the educational sort of things are really important for the museum. So we have an educational group who put on workshops for teachers and also put on workshops for children. So school kids, a lot of big school groups come through. I didn't sort of really show it too much, maybe in that museum zoo, but there's lots of empty space. And that's the thing that I guess a lot of people, you've got really tiny insects and then you've got these huge spaces that you need to fill up. And the images and blowing them up is really good for that. But you also need to have all of those really big areas for people so that when you get these, 60, 70, 100 school kids thundering through, you've got that space for them all to engage and for the teachers to talk to them or the Discovery Centre staff to talk to them. And it needs to be sort of, I guess, so that they can see it. So you need to have little plinths for the kids to stand up on and look at things, or you need to have, a microscope that's set so the kids can see it, or have that image on a screen so that they don't actually have to struggle with the focus or anything like that. So I guess those sort of things, making it really interactive and available to young kids, because we have that's a huge part of our audience. It'd actually be really good to get some more of the older, sort of that mid-teenage to sort of 30 age group coming in, but that's probably the market that we don't have quite so much of. So I guess that's where we'd like to focus, maybe some more digital things or more digital interactives, but I guess that takes money. So to develop all of that, but that's kind of where we'd like to go, I guess, with the new exhibition that we're going to have in 2023.
All right.
Any more questions? Oh, a couple of you have asked, how did you get the spider puppet?
Right, so our prep staff, so we have carpenters and people downstairs who actually put the exhibitions together. So when an exhibition comes in, they actually put it up. And one of them is a puppeteer. And so there is actually, if you go to Webster, Queensland Museum, you'll see a video of the guy behind the puppet. So you'll actually see them talking to him. His name's Chris. And you can see how he developed it, and you know, the puppet's sitting there and he's moving it around. So he's actually developed that. I think it took him about a week to physically make once he had the specimen and the design already. But yeah, there's quite a lovely little video about that.
Yeah, that's
but,
amazing.
yeah, we're very lucky to have him.
Yeah. I guess if there's no other questions, we can wrap up. Are there any more questions from anyone? Okay, thank you again so much, Susan, for your presentation and for joining the symposium. So I guess we'd like to thank all the speakers who made the symposium possible. And I guess at about, do you know what time officially, Jennifer?
In about 15 minutes.
15 minutes,
18
okay.
minutes.
We'll start our meet and greet. And you all can find the link to the meet and greet in the spreadsheet that was shared. Thanks to everyone who attended and participated from the symposium. And then I believe Karen has an announcement as well.
Thank you. So I just want to make one more plug for the tricks of the trade. On Wednesday, we're trying our hardest to make this meeting a little bit more interactive, which is why it'll be in meeting format. And tricks of the trade, you don't have to prepare anything in advance. Unless you want to, you can share your screen and show us a little pre-made video, or you can just talk about what some cool uses of the magic eraser are in the collection, or any other tiny little weird tricks that you might have. It doesn't matter if you invented them yourselves or if you learned them from somebody else. We're just here to share, make it interactive. So we did, I just posted in there a link to a sign-up sheet, and you don't have to do the sign-up sheet. Anybody who wants can just raise their hand, and hopefully we'll have time to get to everybody. But in the meantime, we're going to start with anybody who has put their name up there and just go in order of who puts their name on that sign-up sheet. So if you really want to make sure you got time, make sure you sign up. If not, anybody can just raise a hand, and we'll hopefully get around to letting everybody show their little weird tricks of the trade. So just, sorry, one last plug, and now I'll hand it off to Chris to say goodnight.
Thanks, Karen. Yeah, thank you everyone for these wonderful talks and symposia today. Thank you to the moderators for doing an excellent job. And thank you to the presenters and everyone. It was a great start to ECN. And so tomorrow will be an amazing day. We're going to start at 9 a.m. Pacific, 1600 UTC. Special thank you to all of our vendors and donors. It's because of support that we're able to do things like give 10% of our earnings back to Ento POC and sponsor nearly 40 students for free to join this meeting. And so it's wonderful. We can do that with support from all of our vendors and all of our donors. And lastly, I just want to plug the socials. We've got a social coming up right after this for the Cabinets to the People. So be sure to join that. And then I'm going to host a happy hour after that. Stop by, bring a drink, hang out. Let's see if we can keep Max Barclay up until dawn again. It's wonderful to see everyone, and I'll see you tomorrow morning. Thank you.